guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about anatomy of dicot stem, and if you are new in my channel, please subscribe my channel, and if you like my video, please do like, comment and share my video. So when we talk about dicot stem anatomy, we will say about some layers. So the first layer is the outermost layer, that is this brown layer you can see here. This layer is called epidermis. This is this brown layer. Okay. Now this outermost layer is covered by a thin layer of cuticle. So cuticle is a waxy material and it covers the epidermis. And this epidermis layer contains few trichomes like these, and they also contain few stomata. So trichomes are some hair-like structures you can find in the stem of this dicot plants. Next to epidermis, we can find cortex. So basically, cortex is made up of several layers and it has mainly three zones. The first zone is hypodermis, that is this yellow one that is present just below the epidermis. So this hypodermis contains collenchymatous cells, and this layer provides mechanical strength to the young stems. Next to the hypodermis, we can find this layer, that is cortical layer. This cortical layer contains parenchymatous cells. Next to this cortical layer is the endodermis, this blue one, and here the cells are rich in starch grains, and this layer is called starch sheath. So the function of endodermis layer is mainly to store starch. So this endodermis layer, cortical layer and hypodermis layer, these three layers will make the cortex. Next is pericycle, this red one. So pericycle is placed just below the endodermis. And this pericycle contains sclerenchymatous cells. And the cells in the pericycle layer are found as semilunar patches, just like this. Okay, so next to the pericycle is vascular bundles. So you can see vascular bundles are made up of xylem and phloem. So vascular bundles are arranged in a ring-like structure like this. So this is the specialty of dicot stem. The ring-like structure of vascular bundles is the specialty of dicot stem. And here you can see in each vascular bundle, this purple one is the phloem and the green one is the xylem. And in between the phloem and xylem, you can see the black part. So this black part is called cambium. So in the vascular bundle, you can find three parts. Purple one is phloem, green one is xylem, and in between phloem and xylem, you can get the black one, that is called cambium. Okay, so there are different types of xylem. One is protoxylem, another is metaxylem. So the protoxylem is actually formed first during the tissue formation, and the metaxylem is formed later during the tissue formation. And the protoxylem, you can see here, the protoxylem lies towards the center, that is towards the pith, and the metaxylem lies towards the periphery of this tissue. And this type of arrangement is called endark condition. Endark means the protoxylem is found towards the center and the metaxylem is found towards the periphery. This type of arrangement is called endark condition. And the vascular bundles are called as conjoint since they contain both phloem and xylem together. And vascular bundles are called here open since the cambium is present in between xylem and phloem. Next is medullary rays. So medullary rays are placed between vascular bundles, you can see here. And the next is pith. So pith is the central portion of stem. Here you can find large number of parenchymatous cells in the pith, and they have intercellular spaces. A big intercellular space you can find here between these cells. So this is all about today's lecture. Hope you understood the session. Thank you.